is for freshman biology class. You should have in front of you your protein synthesis part 3 notes entitled Mutations, and we've already discussed transcription and translation. So we're going to continue this lesson with a lesson on mutations, and then we'll be finished with our unit. So let's start off by reviewing the central dogma of biology, which is the fact that DNA can make a copy of itself in the form of RNA, which then is used to make a protein. I've added a couple of details to this diagram just as a matter of reference. You know that DNA polymerase can copy DNA during DNA replication. You know that during transcription, we are making RNA from DNA, and we're using RNA polymerase along with some other helpers. And then in order to do translation, we're taking that RNA and it's being read by the ribosome. And you remember that tRNAs help bring amino acids to the ribosome and that protein is assembled in the correct order. What happens when there is a mutation? If we mutate the DNA, then we will see the effects of that mutation in the RNA, which will trickle down to protein. So in your notes, be sure you get, I've tried to make this easy for you. You can see that I've underlined the things that should go in the blanks. So from here on out, it should be that way. So mutations in DNA cause effects in RNA and in protein. So what exactly is a mutation? A mutation is just a change in the genetic material of a cell. It's a change in the DNA. It could be a change in the RNA as well. Let's say that when you were doing transcription, you accidentally placed a wrong nitrogen base. This happened the other day, or nucleotide rather. This happened the other day when we were doing our lab. Some of you put the wrong nucleotide when you did your mRNA strand and then didn't realize until later you'd made a mistake. So it's totally possible. Anytime there's a mutation, we're going to see some effect. It may not be a huge effect, or it may. It just depends. Also, there's two types of mutations that we'll talk about throughout the school year. We're going to focus today on the second type, but just as a matter of reference, you've heard of chromosomal mutations before. You know that they can cause disorders or death. You've heard of them. You know what Down syndrome is. You've heard of trisomy 21 before. You've probably heard of Edwards syndrome or Patel syndrome. Maybe you don't know exactly what happens with those, but you've heard of trisomy 18 or trisomy 13. These are very devastating mutations to have. Maybe you know someone who's been affected by one of those things. Chromosomal mutations can show up in the form of an entire extra chromosome, like those things I was just talking about, or which is what non-disjunction is, by the way, and we're going to talk about that later, so don't worry too much about that now, but that's what happens when chromosomes don't separate properly, and then of course they're stuck together, so you get an extra, or you could get one too few. Translocation is where big chunks of chromosomes will end up in the wrong place. Maybe a chunk of chromosome 17 ends up on chromosome 5. Inversions are where chromosomes, part of a chromosome would break off and flip upside down. If that happens in the middle of a gene, that could cause a problem. Duplications, where we get an extra copy of a large piece of a chromosome, or if we delete a large piece of a chromosome. So those are all large-scale chromosomal mutations, and we don't really need to focus on how those all work right now. That's really going to come to play in our genetics unit. For now, we need to talk about point mutations. And these are where you just change one base pair. One nitrogen base, one nucleotide is different. Only one. Can that really make a big difference? One single base difference? Yes, it can make a huge difference. It can cause a serious problem, or it might not cause a problem at all. It just depends on where it is and what the effect is. So let's look at some examples. So first of all, there are two different types of point mutations. The first one is the base pair substitution. The second is the frame shift. So in base pair substitutions, we are literally going to substitute a base, one nitrogen base. And within that category, we've got three different effects. So missense is the first effect. And this goes in the blank next to letter A on your notes. A missense mutation is a type of base pair substitution that gives us the wrong amino acid. So here you see the normal DNA template strand, the top one here that's 3 to 5. You see the normal mRNA, and you see our normal protein has 4 amino acids. Now in real life, proteins have way more than 4 amino acids. So this is just an example. You can see met, lys, phi, gly. Down here at the bottom, I'm just going to substitute one letter T for the C. So there's a C here. I've substituted a T. That is going to cause me to have an A in my mRNA strand instead of a G. And because it's right here at the beginning of this codon, it's actually going to cause a different amino acid to show up. And a different amino acid means a different protein fold, which means that protein probably doesn't work anymore. That could be a serious problem. The next type of mutation in the base pair substitution category is called a nonsense mutation. Not only do we get the wrong amino acid, we don't get an amino acid at all, which means we've come to a stop codon too early. So we have a premature stop. This actually happened to one of the groups that during the lab. They got a stop codon really quickly. There should have been 11 amino acids and there were only 2. So again, all I've done is substitute one letter. Should have been a T, it's an A. And I get a U on my mRNA, and UAG is a stop codon. So this protein doesn't even get made. Not only do I have a broken protein, I have no protein. That's a big problem. What if this happened to be the protein that is responsible for one of the steps of cellular respiration? That'd be devastating. Or maybe it's just a protein that says, hey, you should put some brown hair color in this part of your hair. Maybe you've got a blonde spot. Maybe it's not a big deal, but most of the time it is a big deal. All right, and the last one in this category is a silent mutation. There's no effect. Lucky you, should have been a G here. We changed it to an A, which gives me a U on my mRNA. And fortunately, GGC, this codon here, and GGU both give me glycine, the amino acid. So thank goodness that code is redundant. There's more than one codon that gives me glycine. I don't have a problem. Silent mutations are common. The others, not so common. And later we'll talk about why that is. But just think about it for a second. If you have a missense or a nonsense, you have a broken protein or no protein at all, and it's an important protein, are you going to survive? Probably not. If it's hair color, no big deal, right? All right, let's move on to frame shift mutations. A frame shift mutation does just what it says. It shifts the reading frame of the codon over. So look at this normal mRNA AUG, AAG, UUU, GGC, stop. Down here, I'm just going to, instead of substituting, I'm just going to add in an extra A. So my DNA should have read TAC, TTC, but now it reads TAC, and there's an extra A, TT, CAA. It's messed up all of my codons all the way down on my mRNA. Look at that. So if you compare the normal to the mutant, you see all of the codons are wrong. So we have a huge problem. This could result in nonsense or missense. So again, remember, a missense mutation gives me the wrong amino acid. Nonsense gives me no amino acid. So frameshift mutations can be a huge problem, and they're never silent. The second kind of frameshift is a deletion. Same thing. I take one base out, everybody shifts over one, and I get a whole bunch of missense, many, many, many wrong amino acids, or I get a premature termination. Sometimes when protein synthesis is happening, if we end up with so many wrong amino acids in a row, the whole translation process will just stop and the protein will be denatured. All right, here's an example of a mutation that's sort of famous. This is one that you don't die immediately from it. We have medications that can help. In the past, it was very serious. Now it is still serious, but it can be helped. Sickle cell anemia is a type of anemia, which is a red blood cell disorder, where the cells are the wrong shape. And because they're the wrong shape, they can cause some pretty serious symptoms. Pain, frequent infections, delayed growth, strokes, pulmonary hypertension, that's high blood pressure of the lungs, organ damage, blindness, jaundice, and gallstones, just to name a few. Pain and suffering, right? Caused by a genetic defect. It's a point mutation. One single base is wrong. 5% of humans have this gene. And in some places in Africa, you might see up to 25% of people having this gene. And that's because, interestingly, these red blood cells that are misshapen, cannot be hosts to the malaria parasite. And since malaria is a big problem in Africa and places around the equator in general, malaria actually is a little protist organism that lives in the red blood cells and causes, you to be very sick. But since people with sickle cell anemia can't get malaria, they live longer than people with malaria, and they pass the gene on. In the United States, that doesn't happen as much because we don't have the malaria problems. Right, here's how that works. In normal, or we call it wild type, normal hemoglobin, we should have a glutamine residue right here in the middle. One base pair substituted from a T to an A gives me a different amino acid. Just gives me one different amino acid, and I have a huge problem. On the left here, you see the normal hemoglobin. And I know you're not necessarily trained to read this diagram, but you see there's 2 blue chunks and 2 red chunks. And you see these little green things. Those are the heme groups. They carry oxygen. Over here, you can see that instead of blue in the corners and red in the opposite corners, I've got 2 blue and two red. Or orange, I'm sorry. These orange ones are the mutated ones. And that's not going to carry oxygen as well. You don't see as many heme groups, and you also, it's going to cause a big problem in the overall structure. Right here, instead of forming little chunks, it forms a big long chain. So these sticks here are the hemoglobin. They stretch out the red blood cells and don't allow them to slide easily through the capillaries. So up here you can see your normal red blood cells and the normal hemoglobin within the red blood cell. Down here, the sickled cells, they look like a sickle shape, are getting clogged. They're clogging the capillaries. They're not allowing the blood to pass through, and that can result in blood flow problems. Tissue will die, infections, all kinds of things like that. So that's basically what's happening with mutations. We'll be talking some more about this when we get to our genetics unit, but for now, that is it. Thanks for watching. See you later.